This is a thorough discussion of the information in Chapter 25. This is going to go way beyond what you need to know for the test. For the test, look at the posted review questions, answer those questions from the posted review, and you'll be ready for the metabolism part of this exam. There are really three parts to this chapter. Metabolism is one, it's a big one. Nutrition is a second one, and energetics is a third. We're going to kind of bounce and forth a little bit. We're going to go a little bit more detail than we need to, than you need to know for the test. But if you're at all interested in the deep knowledge of metabolism, then we're going to get to that. So we'll talk about nutrients, essential nutrients and molecules. We'll talk about the things involved in metabolic activity, what the processes are, what they're for. Are we making things? Are we breaking things down? And then we'll talk about heat gain and loss in way more detail than you need to know. All right, so to carry out all metabolic reactions, cells require oxygen and nutrients. Oxygen technically is a nutrient, but in our way of classifying things, nutrients are going to include water, vitamins, minerals, and organic substrates, which are things like glucose and amino acids. Metabolism is the sum of all chemical changes that occur in body tissue. Your book says physical changes too, but I don't know what they mean by that. Mostly it's the catabolic reactions which break things down. Think of the word catastrophe and what it means. And anabolic reactions, which are building things up. If you're making protein, if you're storing fat, then you're doing anabolic reactions. The nutrient pool is everything that is available. In general, you think of the nutrient pool as being in the bloodstream, but you store some of that in your fat reserves, you store some in your liver. So here's a great picture that shows you how that nutrient pool is sort of divvied up. You've got the food from your diet at the top. And we're only going to talk about the three major nutrient categories: fats, carbohydrates, and proteins. Fats get broken down to free fatty acids and glycerol. They're going to be put into your fat stores through a process called lipogenesis, which is making fats from the free fatty acid pool. If you have excess glucose, look at the middle, from the carbs in your diet, that glucose can shift into the fat reserves through lipogenesis. And then into the glucose pool and into glycogen stores, which are going to be in your liver and skeletal muscles through glycogenesis. Glucose mostly feeds the brain. If there's any left over, it can be used in most tissues. But in general, look down at that metabolism in most tissues. Little vat at the bottom on the left, and you see that most tissues are fed free fatty acids. If there's excess, they kind of circulate back in. Look back at that glucose pool in the middle and notice that if it drops too low, only the brain gets to have glucose. Everybody else has to feed off of free fatty acids. And then on the right, proteins are, of course, broken down to amino acids. You've got proteins in the body through protein synthesis, and you have an amino acid pool, but notice that that's connected to the glucose pool. And by the way, lipids can do this as well. It's a process called gluconeogenesis, which means literally making new sugar. And when you turn amino acids or fats into sugar, that's gluconeogenesis. All right, so our first category is the catabolism or catabolic reactions. This is going to take large molecules and break them down into smaller ones. So if you take glucose and break it down to carbon dioxide, you're going to release energy that can be used to make ATP. Anabolic reactions, anabolism, is going the other way. It requires an input of energy, so you're making large molecules out of small ones. So if you're storing glucose as glycogen or storing fats, then you're doing anabolic reactions, and they require energy. And you get that energy from the catabolic reactions that produce the ATP. Okay, we have nutrient reserves. Triglycerides are your fats, and that's going to be stored in your adipose tissues, which you know are scattered throughout the body. Those are going to be liberated by fatty acids. So you're going to have fatty acids in the bloodstream to feed your body tissues. Glycogen, which is the way we store glucose. Glycogen is stored in two, and as far as I know, only two tissues, and that's liver and skeletal muscle. And then, of course, proteins, and proteins are made when they're needed. You don't store extra protein. You just make the proteins your cells need. That's regulated hormonally. And even though they're abundant in the body, still, you're not just going to stockpile extra protein. So, energetics is the study of the flow of energy and as it changes from one form to another. And there's a primary kind of reaction, actually a pair of reactions, called oxidation and reduction. Oxidation in biology is losing hydrogens or electrons, and reduction is gaining. Now that sounds backwards because reduction should be losing, shouldn't it? Well, it's called reductions because electrons have a negative charge. So if you give electrons to a molecule, you're reducing its electrical charge, and that's why it's called reduction. In oxidation and reduction reactions, which have to go together, you can't oxidize one thing unless you have something else ready to be reduced. Those electrons that you're moving from the thing you're oxidizing to the thing you're reducing carry chemical energy, which can then be harvested, at least some of it can be harvested, and that can be used to make ATP. So when you reduce things, remember you're adding electrons. Those electrons are carrying energy. So things that are reduced gain energy, things that are oxidized lose energy. And since those electrons are generally tied to hydrogens, organic molecules with lots of hydrogens have lots of energy, and organic molecules or even inorganic molecules that don't have a lot of hydrogens don't have a lot of available energy. So how do we get that energy out of those electrons? Well, in the mitochondria, in the membranes inside of the mitochondria, there is a special set of proteins and related chemicals called the electron transport chain. And electrons are passed through this as a series of oxidation-reduction reactions. And eventually those electrons and the hydrogen ions that go with them are transferred to oxygen, and that oxygen is converted to water. That's the way you get rid of them at the end of things. And we're going to see that in more detail. So if you just have the basic idea that in oxidation reduction, you're stripping electrons off of something you're oxidizing, and you run it through this transport chain to harvest the energy out of it in a way that we'll see. But after you've harvested the energy, those electrons and their associated hydrogen ions have to go somewhere, and that's the entire job of oxygen. Oxygen is the final electron acceptor in oxidation-reduction reactions in mitochondria. All right, well, enzymes can't work in a vacuum. They need some coenzymes. We're going to see there's a number of important coenzymes. Technically, ATP is a coenzyme. Coenzymes are organic molecules that are carrying something or assisting in some way. ATP is a coenzyme because it carries high-energy phosphates from one reaction to another. NAD and FAD carry hydrogens. They are going to transfer those hydrogens and their electrons. They're going to be oxidized and then reduced. So they are transporters of hydrogen from one chemical reaction to another, from one part, say the cytoplasm or inside the mitochondria, to get those hydrogens to the electron transport chain. FAD, which is made from the B vitamin riboflavin, can carry two hydrogen ions and two hydrogen atoms, the hydrogen ion plus the electrons, and form something called FADH2. In other words, it's just combining with two hydrogens. NAD, which is formed from the B vitamin niacin, in its oxidized form has a positive charge, so even though it picks up two electrons and forms NADH, there's an extra little hydrogen that's going to kind of go along with it. So each of these technically can move two hydrogens, that's two hydrogen ions and two electrons, from one set of reactions to another. Most of our time is going to be spent on carbohydrate catabolism. We'll be looking a little bit at anabolism. Remember, that's building up. We'll also be looking at lipid metabolism and protein metabolism. But most of what we eat, the main energy molecule that we eat in our diet is glucose. And that glucose comes to us in the form of starch, which is made by plants, or glycogen, which is found in meat, which is nothing other than animal muscle. So glucose is the primary fuel molecule we eat, therefore it's the primary fuel molecule that we break down. So when we look at the respiration of glucose, we're talking about cellular respiration, which is extracting the energy out of the glucose by combining it with oxygen and generating carbon dioxide and water. You've already covered the respiratory system, so you already know external respiration is gas exchange, CO2 and oxygen exchange at the lungs, and internal respiration is CO2 and oxygen exchange at the tissues. So here, cellular respiration is just the exchange of oxygen and CO2 in the cells. So that's basically what is happening with respiration. We're going to take glucose, combine it with oxygen. We're going to strip all the hydrogens out of that glucose, oxidize it completely to carbon dioxide, take those hydrogens, stick them on oxygen, and that's going to make water. All right, we've got 3 basic steps. We're going to do glycolysis, the citric acid cycle, and electron transport. In the human body and in most organisms, glycolysis occurs in the cytoplasm of the cell. In eukaryotic organisms like us that have mitochondria, citric acid cycle and electron transport occur in the mitochondria. The amount of ATP you can get from one molecule of glucose depends on how you calculate it. Your book says 30 to 32. Most people will say 36 to 38. So it just all depends on how they're counting those ATPs. And what we're going to see is it's not an exact number anyway, but it's 30-something. All right, so our first step is glycolysis. This occurs in the cytoplasm of the cell. And glycolysis is not the part that requires oxygen. The part that requires oxygen is in the mitochondria. We're going to take glucose, which is a 6-carbon compound, and we're going to energize it, and then we're going to pull some of the energy out of it. We're going to wind up with 2 3-carbon compounds, molecules called pyruvate or pyruvic acid. And pyruvate still has some energy in it because it still has hydrogens in it. So when we finish glycolysis, we're going to get a little bit of energy out, but then we're not done yet. We are going to finish metabolizing the pyruvate in the mitochondria, but glycolysis stops with pyruvate. It starts with an enzyme that adds a phosphate, a high-energy phosphate to glucose. This creates an intermediate step called glucose 6-phosphate, and it requires glucose, of course. It requires the enzymes. It requires ATP, some inorganic phosphate groups, and we are going to see we need some NAD. So here are the energy requiring steps. And the energy requiring steps, there's 3 of them, notice. Just look at the pictures first. We're going to, at step 1, take a high energy phosphate from ATP and put it on one end of glucose. In the second step, we're going to take a second high energy phosphate from ATP and put it on the other end. In the process of doing that, we've converted glucose to fructose, and it's got 2 phosphates, one on carbon 1 and one on carbon 6, fructose 1,6-bisphosphate. And then, at the end of this process, we're going to split it in half. When we split it in half, the 2 halves initially are not the same, but notice the one called dihydroxyacetone phosphate is going to get converted to the glyceraldehyde 3-phosphate. Listen, there are other names for these things. Don't get bogged down on the names. Just look at the overall process. So at this point in glycolysis, we put in the energy from 2 ATPs. That energy is still there in the form of those energized phosphate. And then when we broke it in half, we have 2 3-carbon compounds, each with a phosphate on one end. So what we're going to do next is try to harvest that energy out. So once we have our 2 little 3-carbon guys, we're going to take some hydrogens off in the form of NADH. So we're going to take NAD and convert it to NADH. We do this twice for each glucose because we had 2 of those little 3-carbon compounds. We're going to add in some inorganic phosphates, which just by pulling the hydrogens off of our little 3-carbon guy, when we put those hydrogens on the NAD, it released enough energy to energize those phosphates without needing additional ATP. So now in the next step, which is step 5 in the picture, We're going to harvest some energy out. We're going to make ATP. And in the next step, we're going to pull some hydrogens out and we're going to rearrange it. In step 7, we're going to pull out 2 more ATPs, and that's going to give us pyruvate. So the bottom line is we took 1 glucose, we put 2 ATPs in, we pulled 2 NADHs out, we pulled 4 ATPs out for a net gain of 2 ATPs, and we wound up with 2 molecules of pyruvate, and that's the end of glycolysis. And remember, that occurs in the cytoplasm. Now, as we move on into what we call aerobic metabolism, this occurs in the mitochondria, absolutely requires oxygen. If you don't have oxygen, this whole process it's like there's a jam in the works. It just stops. Everything in the mitochondria stops without oxygen. If it's a brain cell you're talking about, the cell simply dies without sufficient ATP. Some cells can manage without oxygen. In humans, that's our skeletal muscle cells, and we'll see how we do that. But other cell types, heart, brain, liver, kidney, they're just going to die if oxygen is not available. What we're going to do in aerobic metabolism within the mitochondria is release the remaining energy that's in that pyruvate, and in the process, we're going to get a lot of ATP, a lot more ATP than we got in glycolysis. There are 2 steps inside the mitochondria, the citric acid cycle and the electron transport chain. And to appreciate how this is all going to work, we need to look briefly at the structure of the mitochondria. You remember the mitochondria from AMP1. It has a smooth outer membrane and a very highly folded inner membrane. And that outer membrane and inner membrane have a space between them. Look at the bottom. We just call that the intermembrane space. The outer membrane has to have openings in it large enough to let pyruvate in and NAD in and out, NAD and NADH, ATP and ATP in and out. It says they're pores. They're actually probably transporters. So there are transport proteins in the membrane. Not just in the outer membrane, but connecting the outer membrane to the inner membrane. They're transporters. We have to get to the inside of the inner membrane, and that space inside is called the mitochondrial matrix. So what we're going to see is that there are kind of 2 regions, actually technically 3. The matrix is the very deep interior of the mitochondria. The inner membrane itself, with all of its folds, is going to do our electron transport. And it's going to use that intermembrane space between the outer and inner membranes. So as we get to each of these parts, I'll tell you where they are. So in the citric acid cycle, we're in the matrix. We're inside of that inner membrane, just sort of dissolved. There are enzymes there. There's NAD there. There's FAD there. The pyruvate is going to come in there. And the enzymes and the coenzymes are going to strip the hydrogen atoms off of the pyruvate. And those hydrogen atoms, remember, are carrying energy. So we're going to have to deal with those hydrogen atoms, and we're going to do that with the electron transport chain, which is the 3rd step. So in the citric acid cycle, we're pulling the hydrogens off, and what we're leaving behind is just the carbon and oxygen, and that's going to get converted to CO2. So in the citric acid cycle, we pull off all of the hydrogens off of the carbon, leaving behind CO2, which you know is a waste product, and it can just diffuse right out of the mitochondria, out of the cell, into the venous blood circulating, and get released at the lungs. Okay, so pyruvate has to interact with a couple of things to start with, and they don't mention that one of those things is an enzyme, but pyruvate interacts with NAD and a coenzyme A and an enzyme which strips some hydrogens off, so we get the NAD converted to NADH. The coenzyme A picks up the remains of the pyruvate. We're going to strip a CO2 off. Remember, pyruvate had 3 carbons, so we're pulling one of those 3 carbons off as CO2, and that leaves us with acetyl-CoA, an acetyl group bound to coenzyme A, and that acetyl group has 2 remaining carbons from the original pyruvate. So this gets carried to the citric acid cycle itself. The acetyl group gets combined with the 4-carbon molecule oxaloacetate, and oxaloacetate plus the acetyl group makes a 6-carbon citric acid, which is why this is called the citric acid cycle. The coenzyme A is released. It's just going to go back and do the same thing over and over again. It just brings the acetyl groups from the pyruvate reaction into the citric acid cycle. One citric acid cycle removes the 2 remaining carbons from the acetyl group, and it regenerates that 4-carbon chain. And there are a lot of steps involved, and there are more hydrogens being pulled off, and there are other things that are happening. So one citric acid cycle produces not just 2 molecules of CO2, it releases at one step enough energy to trap another high-energy phosphate. Now, don't ask me why, but it's gonna form GTP. That's just guanosine triphosphate, just like the G and the A of DNA is the same as the A in ATP is adenosine from RNA, and then it's going to just hand that high-energy phosphate off to ATP. So you might as well just say it produces 1 molecule of ATP. That summary is too complicated. We're not going to look at it in too much detail, but I do want you to pick out how many NADs and FADs are actually going to be produced in one cycle of the citric acid cycle. So 3 NADH and 1 FADH2, and that's going to go around twice for each glucose. So per glucose, we're going to get 6 NADH and 2 NADH2 plus 2 ATPs. All right, so here's the picture that depicts everything that we just talked about. So we're starting with pyruvate. Remember, we're in the matrix of the mitochondria. We add in an NAD. It picks up the hydrogen, becomes NADH. And if you follow the orange arrows from NADH, all the NADH is going to go to the electron transport chain. You're dropping back up at pyruvate. You're adding in coenzyme A. You're dropping off a CO2. That acetyl-CoA goes to combine with the 4-carbon oxaloacetate. It then forms citrate. And notice we've got a number of steps. Your book is leaving out just about all the steps. The hydrogens, though, are going almost completely to NADH. The CO2s, you can see 2 more CO2s coming off. So we're done with all the carbons and oxygen we got from the pyruvate, and all that's left to deal with is the hydrogens. So we're going to make 2 CO2s. We're going to make a total of 3 NADHs, an ATP, and an FADH2. Here you see it in a little bit more detail. This is way too much information, but if you're interested in following all the steps, you can Google videos or go to YouTube and watch videos of this process. And in fact, I gave you the video link for your PA assignment that went through the Krebs cycle. So here's more detail. Again, please just review to what's in your review questions posted in Canvas so you know how much detail you need to know. Okay, so now we have all these hydrogens that we're carrying in NAD and FAD. And so we're going to move to the inner membrane of the mitochondria where we're going to do oxidative phosphorylation, which means we're going to use oxygen to make ATP. We're going to put the phosphorus, the phosphates, onto the ATP using oxygen. So we have this sequence of electron carriers, and I'm going to take the hydrogens when I'm done with it and combine them with oxygen to make water. Okay, so the electron transport chain is a series of proteins in the inner mitochondrial membrane, and we're just going to think of them as these 4 steps connected by 2 electron carriers, okay? So the protein complexes, the respiratory protein complexes, one of the electron carriers is coenzyme Q, and you've got some cytochrome molecules. All of these things are just like playing hot potato with these electrons. And again, this is just more information than you need at this step. Let's just go through the steps in order. So our first step, we're just going to call respiratory complex 1. And respiratory complex 1 has really 2 things that it does. It takes the hydrogens just from NAD, from all those NADs, the NADH, takes those hydrogens off, and it passes the electrons from those hydrogens to coenzyme Q. So it's transferring the electrons, but it also releases enough energy from those electrons to pump a hydrogen. That hydrogen came from the NADH. It's a hydrogen ion, and it moves from the matrix into that intermembrane space. So step 1 is NADH, loses its hydrogens. Step 2 is kind of the same thing, except for the FAD. In respiratory complex 2, the hydrogens from FAD are picked up. The electrons are shuffled off to coenzyme Q. Notice, though, that we're not releasing enough energy here to pump any hydrogen. So that's something that complex 1 can do that complex 2 can't do. In step 3, we're just going to go to complex 3, and it doesn't matter whether the hydrogens came from NADH or FADH. They all went to coenzyme Q. CoQ can transfer those electrons to respiratory complex 3. And in the depths of respiratory complex 3, the electrons are passed along, and enough energy is released to pump another hydrogen into the intermembrane space, and then the electrons are going to get passed off to complex 4 through the cytochrome chain. In step 4, respiratory complex 4, we're going to take the rest of the available energy to pump one more hydrogen into the intermembrane space. The electrons then in complex 4, through an enzymatic reaction, combine with oxygen to produce water. Okay? So if we go back through those steps again, step 1 took the hydrogens off of NADH and pumped a hydrogen. Step 2 took a hydrogen off of FADH2. It did not pump a hydrogen. Whether it's step 1 or step 2, those electrons get passed to CoQ. In step 3, from CoQ to respiratory complex 3, we pump another hydrogen. And then in step 4, we pump a third hydrogen. Remember, it will be only 2 hydrogens total if you started with FAD. And we split oxygen and combine those hydrogens to make water. Okay? So why do we do all this? Why are we doing all this pumping of hydrogen? Well, what we're doing with all those hydrogens is we're collecting them and storing them in the intermembrane space. So we have a really high concentration of hydrogen in that little tiny intermembrane space. And there we're going to park our most important part, which is an enzyme called ATP synthase. And ATP synthase uses a process called chemiosmosis. Chemiosmosis is the movement of those hydrogen ions back from the intermembrane space through the inner mitochondrial membrane back to the matrix. And when those hydrogens channel through the ATP synthase, that powers the production of ATP. So here you see the diagram in your book. Honestly, I know you can't see this. Let's just look at a close-up of the membrane. So here again, we've got the matrix at the bottom. We have the intermembrane space above the inner membrane. Follow the NADH from the left going through complex 1, FADH2 going through complex 2. And whether the electrons come from complex 1 or complex 2, the electrons go to coenzyme Q. From Q, they go to complex 3. From complex 3, they're going to go to something called cytochrome C. From there, they go to complex 4 and follow the electrons into the matrix. They're given to the oxygen, and that combines to make water. All right, let's start back at NADH. Follow the hydrogens this time. The hydrogens are released in the matrix from NADH and FADH2. If you start with NADH, you have enough energy to pump a hydrogen through complex 1. You pump a second hydrogen through complex 3 and a third one through complex 4. And all of those hydrogens are just kind of just clustered in that little tiny intermembrane space, giving you a very high concentration of hydrogen there. And then notice the ATP synthase on the far right. It's got a proton channel, a hydrogen ion channel that lets the hydrogens back through. And as they funnel through there, they release enough energy to produce an ATP. The numbers of ATP produced per hydrogen is hard to calculate. It's actually not a set number. But in general, what we say is that each NADH releases enough hydrogen or pumps enough hydrogen to produce 3 ATPs, and the FADH2s can only produce the equivalent of 2 ATPs. So the hydrogen ions get pumped into that intermembrane space from complex 1 and complex 3 and complex 4. And then the NAD is responsible for pumping 6 hydrogens, the FAD for 4 hydrogens. So you see it's not the amount of ATP being produced. 2.5 molecules, how do you get half a molecule? Well, of course you don't really. So it's not an exact number. It's just the amount of energy that you produce. And so the grand total of oxidative phosphorylation, let's see, we had, for oxidative phosphorylation, we had 2 NADHs from glycolysis. We had 2 from converting pyruvate to acetyl-CoA. We had 3 NADHs from the citric acid cycle, so that's, go through that twice, so that's 10 total NADH, which gives us close to 30 ATPs. And then we had 2 FADH2s, so that's about 4 more ATPs, so we're up to 34. And then we had 2 ATPs from glycolysis and 2 ATPs from the citric acid cycle, so we've got about 36 ATPs. Okay, so if you start with glucose and you end with carbon dioxide and water, you've gone through that whole process, you get the equivalent of between 32 and 36 ATPs. All right, so just kind of going through it all again, and of course you're welcome to rewind and re-listen to the video. 1 glucose molecule got broken down without using any oxygen to 2 pyruvates. We got 2 ATPs in the process plus 2 NADHs. And the NADH is going to go into the mitochondria. And then in the citric acid cycle, we have to go through it twice because we got 2 pyruvates for each glucose. We got 1 ATP directly from the citric acid cycle, so that's total to 2. We transferred hydrogen atoms to, let's see, there was an NADH in the conversion of pyruvate to acetyl-CoA, and then 3 of them in the citric acid cycle itself. And then for each molecule of glucose, we get a total of 10 NADH and 2 FADH2. Your book is trying to be really accurate by saying 2.5 ATPs. I just think that's kind of overkill. So somewhere in the vicinity of 32 to 38 ATP, depending on how you're counting them. Don't have to remember it exactly, but you do have to remember 2 from glycolysis, 2 directly from the citric acid cycle, and more from the NADH than from the FADH2. So 30-something in the mitochondria in oxidative phosphorylation. So here's this nice overview in your book. Take your time going over it if you need to. Refer to this to answer the questions on the review and to make sure you understand the material. All right, so that's it for the aerobic respiration of glucose. Now we're going to go into some anabolic pathways. Sometimes your blood glucose level falls too low, and you need to feed glucose to your brain. So if you don't have enough glucose in your blood, your liver is going to release what's stored, and you're going to make new glucose, gluconeogenesis, from either amino acids or from the breakdown of fats. Glucose stored as glycogen is not really gluconeogenesis. Glucose stored as glycogen is just broken down through a process called glycogenolysis, and that's going to increase your blood glucose level. And I know I haven't talked about the metabolic states yet. That's going to come later in the book, and we'll tie it all together. So glycogenesis is making glycogen. You do that when your blood glucose level is high after a meal. Glycogenolysis is the breakdown of glycogen. You do that when your blood sugar is low. And that glycogenolysis can occur in liver and skeletal muscle, but to feed glucose to your brain, you're going to rely on liver. The glycogen in your skeletal muscle is there to feed your muscle cells. Okay, so once again, you have this nice overview. Notice the directions of the arrows. Be sure you can tell which one is anabolic and which one is catabolic so that you can keep track of, am I storing it or am I breaking it down? In the right-hand side, you see other carbohydrates can feed into glycolysis itself. Glycerol, which is produced from fatty acids, can also feed into glycolysis. So there's lots of metabolic pathways involved here. Okay, so this is just showing us a close-up of where in the mitochondria your NADH is being released. Look, though, and see that some amino acids can get converted to pyruvate, and some other things can also be kind of interconverted here. So you see lactate on the left. Lactate is produced in your skeletal muscles when you run out of oxygen. And so pyruvate can get converted to lactate in skeletal muscles when you're actively exercising, but then the lactate can go backwards to form pyruvate once you're done exercising, either in the muscle or in the heart cells or in the liver cells. Okay, lipid metabolism. We've got to be able to make lipids and break them down. So the lipids that we metabolize are almost always the triglycerides. These are the fats and oils in your diet, but in your body they are the fats that you store. So assuming you ate some fat in your diet, you're going to digest it. That's lipid catabolism, breaking it down, lipolysis. If it's been a while since you ate and you're relying on your fat stores, you're going to do lipolysis in your adipose tissues and in your liver. You're going to break those triglycerides down to glycerol, which can be converted to pyruvate, which can go into the citric acid cycle. And the fatty acids themselves can get converted directly to acetyl-CoA. So in beta oxidation, what you're doing is you're taking fatty acid molecules. Remember, these are just long chains of carbon and hydrogen. They're 14 to 28 carbons long. You just break them, chop them into 2 carbon fragments, and feed them directly in as acetyl-CoA. So each one of those 2 carbon fragments is going to have 1 or 2 hydrogens on it. So you're going to get acetyl-CoA. You're going to get a lot of NADH, FADH2, and release the CO2 that's left over. All right, so let's take a look at the picture. So at the top, you can see a triglyceride molecule. That's a fat molecule. You absorb it into a typical cell through endocytosis. It's going to get broken down. Notice the glycerol goes straight down. The glycerol, that is the part at the far right in the upper picture that has all the oxygens on it, gets converted to pyruvate. And that generates a couple of ATPs. And then the pyruvate goes into the mitochondria and does what it always does. The fatty acids, when they're broken off, each fatty acid, look over on the left at beta oxidation. Each fatty acid is just chopped off 2 carbons at a time. And you feed that into the citric acid cycle. So lipids carry a lot of energy, more energy gram per gram than carbohydrates do. So from one 18-carbon fatty acid molecule, you can get 120 ATPs. That is huge. The amount of energy stored in fats is huge. So it makes sense that when we have extra calories in our diet, we store them as fats. They're lighter in weight, and they are just really packed with energy. So when you make lipids, when you have an excess of calories in your diet and you're making lipids, you're doing lipogenesis. And you can do that not just with lipids, you can interconvert other substances, including amino acids. So if you eat too much protein or too much carbohydrate, your liver does this interconversion and it stores everything as fat. So if no matter what the source of excess calories in your diet, yeah, you're going to convert it to fat. Okay, so there are some issues here. Your liver is not able to make all the fatty acids that you need. There are some essential fatty acids that are used for specific metabolic pathways. They may be involved in making signaling molecules or specific fatty acids needed in plasma membrane synthesis or myelin synthesis. So here's a term you need to know: essential versus non-essential. Essential means you can't make it or you can't make enough of it, and therefore you have to get it in your diet. So there are lots of non-essential fatty acids, and we can make all the steroids we need, so they're non-essential. Steroids, of course, include cholesterol. You do not need to know which fatty acids are essential. Your book gives you the examples of linoleic and linolenic. I think there are only 3 total essential fatty acids. But what that means is you've got to make sure you get enough fat in your diet to make sure you're consuming the essential fatty acids. So if you went on a totally fat-free diet, you would be malnourished. You would not be getting the nutrients you need. You store lipids, of course, in your adipose tissues. You also store some lipid in the muscle cells of your heart and skeletal muscle, and you store excess fat in your liver, which can cause problems if you have an excess. There are lots of pathways to make lipids. If you over-imbibe alcohol, for example, alcohol gets broken down to acetic acid, acetyl groups, and gets converted to fat in the liver. You also have to be able to transport lipids because they're not soluble in water. So just getting lipids from your digestive tract to your liver, from your adipose to your body cells, it's not that simple. It's not that straightforward. So what we've got to do is we've got to package all of our lipids, not just our triglycerides for energy, but our steroid hormones, our fat-soluble vitamins, the phospholipids our cells need to make their membranes. All of these have to be transported in the blood and distributed throughout the body. So to do that, because they're not soluble in water, we package them in protein. So we kind of wrap a protein wrapper around our little lipid bodies, our little collected assortments of lipids, and we call these lipoproteins. And there are different kinds of lipoproteins. And yes, you have a little bit of free fatty acid dissolved in your plasma, but not a whole heck of a lot. The free fatty acids, though, are really mobile. They can get in and out of the bloodstream very easily. They're generally pretty small fatty acids. Albumin is the main plasma protein, y'all remember that, and it can pick up some fatty acids. And those fatty acids are just either diffusing out of your lipid reserves, or remember, you can't hold them in cells in the plasma membrane, so they might have just gotten absorbed through your gut. So they're just free fatty acids. Free fatty acids can get into cells, typical body cells, heart cells. Any organ except the brain will metabolize free fatty acids. So when you're sitting there at rest and it's been a while since you ate and your blood glucose is pretty low, your cells are living off these free fatty acids. Everybody except your brain. Okay, so on to our lipoproteins. You've got different kinds depending on the metabolic state of your body and what you ate and just part of it's just genetic. But these lipoproteins are just these big clusters of lipid molecules. It's going to have triglycerides, it's going to have cholesterol, it's going to have the fat-soluble vitamins, anything else that's lipid. And it's got kind of a wrapper of protein around it. And we have four groups: chylomicrons, VLDLs, LDLs, and HDLs. And we're going to talk about each one of those in turn. Chylomicrons are what you put into the bloodstream from what you ate. In other words, you digest fats in your diet, as we've already seen. Those digested fats get into the intestinal lining of the small intestine, the epithelial lining. Those cells produce these things called chylomicrons. They repackage all the fats. They add in the cholesterol and the fat-soluble vitamins, and they wrap it up in a protein. And that's a chylomicron. And chylomicrons are too big to get into the bloodstream. So they're going to go into the lacteals. Remember, those are the lymph capillaries of the intestinal villi. The lymphatic system carries them to the thoracic duct. They get dumped in at the left subclavian vein and out into the general circulation, where they can feed your body cells. So here in this picture, you see them being absorbed as micelles. We studied that last chapter. Repackaged into chylomicrons, picked up in the lacteals, into the general circulation. And look at step 4. Your blood vessels have an enzyme, lipoprotein lipase, which can break down those chylomicrons, release the fatty acids, and those then get absorbed into your cells. Your resting skeletal muscles can absorb them and then just store them as fat reserves. Your adipose can pick them up. So those chylomicrons are directly feeding the cells of your body that are capable of picking up fatty acids. That includes the liver. It doesn't show it in this picture because the liver has other functions. So here we see chylomicrons coming into the liver, and then the liver cells are actually going to repackage what's left of those. So what's the remnants of the chylomicrons get taken in by your liver cells, and they repackage those lipids with a new protein wrapper as a VLDL. And VLDL's job is to grab any remaining triglycerides and deliver them to the muscle and adipose. And going back to the last picture, that lipoprotein lipase in those capillaries of your skeletal muscle and your adipose tissue will pick up those fatty acids and store them. So that's the job of VLDL. The LDLs, and sometimes the LDLs may just be the remnants of the VLDLs, but LDLs are going to carry a lot of cholesterol, some fat, but mostly cholesterol and protein. And LDL is low density lipoprotein, and VLDL is very low density. The LDLs, look, they actually get eaten, endocytosed by peripheral cells. And peripheral cells, again, that just means just about anything. So you're not only feeding fat to these cells, you're providing them with the cholesterol they need, and they can even break down the protein and use that for their own protein manufacture. So LDL is the way the liver feeds your cells. Now, if those cells can't use all the cholesterol that was in those LDLs, then the cholesterol gets released to the blood. So we're down at step 11. Cholesterol not used by cells re-enters the bloodstream. So look at what else your liver is doing. It's releasing something called an HDL. And HDL you should think of as it's just like an empty truck. And its job is to pick up any excess cholesterol and deliver it back to the liver. So HDL removes excess cholesterol from the bloodstream. And the whole LDL, HDL balance in the body is of concern, because if you have an excess of cholesterol, then you can get atherosclerosis, which can lead to cardiovascular disease. So what you want is to maximize your HDL and minimize your LDL. You don't want to get rid of LDLs. You're feeding your cells through LDLs. But if you have more fat than you need, then your liver's going to produce more LDLs than you need to have. And so your LDL level can get too high. Some things that can contribute to this, unfortunately, genetics can. Diet, absolutely, if you have too much fat in your diet. But even some conditions, for example, people that are hypothyroid very often have a high LDL. HDL, likewise, genetics plays a role, but actually exercise can increase your HDL level. So if you have low HDL, you need to watch your fat intake, but you also need to exercise more. Okay, so that's kind of the story on lipid transport. So now we're going to move on to protein metabolism, and we're not getting bogged down in how do you decide which proteins to make and making different proteins in different cells, because that's more about genetics. How does each cell regulate what proteins it makes? So we're just looking at an overview of how does your body make the proteins that it needs. And we already know that proteins are built from 20 amino acids. We know what their functions are. But how the body decides to make protein is not related to how much protein you have in your diet. So if you eat more protein than you need, then your liver cells are going to catabolize those amino acids. There are a couple things they can do with them. They first have to pull the amino group off, but then some amino acids can go straight into the citric acid cycle. Some amino acids can get converted to pyruvate, and some amino acids can get converted to acetyl groups. So you can feed amino acids into either glycolysis or the citric acid cycle directly in liver cells. Kidney cells to some extent, but most of your body cells are not going to do this. The problem is you've got to get that amino group. And that amino group is the NH2 that all amino acids have. And this is where you need vitamin B6. It's a coenzyme to pick up that amino group. When you pull that amino group off, you can do a couple of different things with it. You can stick it on a different substance to make a new amino acid. Now, you need 20 amino acids, but 8 of them are essential amino acids. And if you remember what that means you have to eat them. So if you're not getting complete protein in your diet, you may be malnourished because you're not getting one or another of those essential amino acids. But for the 12 non-essential amino acids, your liver can transaminate. That is, take an amino group off of one amino acid, stick it on a different organic acid to make another amino acid. So transamination is just transferring an amino group from one little molecule to another. Deamination means you're just taking the amino group off and you're going to throw away what's left. You're going to deaminate amino acids that you're going to metabolize through the citric acid cycle or through glycolysis. The problem with those amino groups is that they're kind of toxic. They make ammonia, and ammonia we know is a base. And if you get too much ammonia accumulating in your body, then you have an acid-base imbalance. You become alkalotic. You have too much base, alkalosis. And so what the liver does with these amino acids is it combines them with a CO2 to make urea. This is called the urea cycle. And urea is still toxic, but it's not nearly as toxic as ammonia is. So consequently, the liver makes urea as a way to get rid of ammonia, and the ammonia is the remains of that amino group. So transamination is simply converting a keto acid into an amino acid, and that's just a way to make more copies of an amino acid you need for protein synthesis. Deamination is pulling the amino group off, but it creates ammonia, and then we're going to, there's our transamination, there is our deamination, and we're going to take that ammonia and convert it to urea. This is fairly harmless. If it builds up, it causes problems, but it's not nearly as bad as ammonia. People with end-stage liver disease aren't very good at this step, and consequently, you have to curtail the amount of protein in their diet, or else they release too much ammonia, and they're not good at making the urea, and they get into a situation where their pH is too high, alkalosis, which causes seizures and convulsions. Okay, so here's the urea cycle. We're going to take 2 ammonias and a CO2 and make a urea out of it. That urea can go into the bloodstream. It's going to go to the kidneys and get excreted as urine, which is how it got the name urea to begin with. Okay, so the liver cells can break down excess amino acids from your diet, but if you're on a strongly curtailed diet, you're just not eating enough calories, then you can start breaking down body proteins, and not just in your liver, but also in your muscle cells. So liver and muscle cells will break down their internal proteins, send those amino acids to the liver. The amino acids are deaminated, and they're going to get thrown into either glycolysis or the citric acid cycle to produce ATP. This is why a severely calorie-restricted diet is really not a very healthy way to lose weight, because when you lose a lot of weight quickly through just not eating enough, you break down protein. And then generally speaking, people who go off those diets, they fall off the wagon, so to speak, they actually start eating too much food, and you tend to store that as fat. So if you diet, gain weight, diet, gain weight, diet, gain weight, you tend to lose protein over time and gain fat over time. So generally speaking, your proteins are not going to be your first choice for breaking down just for generating ATP, because that ammonia is toxic, and because proteins are important to cells. But you will if you're restricted calories too much. Okay, so protein synthesis occurs in cells when they need them. Here are the essential amino acids. You don't need to know which ones they are, versus the non-essential ones, which we can do by transamination. So here you can see taking alpha-ketoglutarate, which is an organic acid that's in the citric acid cycle, add an amino group to that, and you have glutamic acid, which is one of the amino acids. Again, you don't need to remember that detail. Okay, finally we get to the metabolic states, the absorptive and post-absorptive states. And we're going to put together everything we've learned about metabolism so far as we go through these states. So the tissues we're going to be talking about: liver, adipose, skeletal muscle, nerve tissue, and then everybody else. So I think the first thing you need to know is what the absorptive and post-absorptive states are. Absorptive state occurs when you have food in your gut. You're absorbing nutrients from your diet. So the main source of nutrients getting into the bloodstream is actually from your intestine. You're absorbing your dietary nutrients. And the post-absorptive state comes when your gut is empty, hours after you finished eating. So the liver, of course, is the most important organ in regulating metabolism during these two states. And the liver is regulated itself by hormones, which either turn on or turn off enzymes or make nutrients available. So we studied the liver last chapter, but when we do most of the metabolism in the liver, we're using just the main kind of liver cell, the hepatocyte. Remember, the other kind of liver cell is the macrophage, and they have more of a role in protecting from infection or getting rid of old red blood cells. But every metabolic process that we say this is done in the liver, almost always it's the hepatocytes that are doing that. And one of the things they do is they store stuff for you. They store not only glycogen as a reserve of glucose, they're going to store vitamins for you, and they store lipids for you to some extent. Most of the lipid storage, of course, is going to be in the adipose tissue. And adipose tissue, is scattered throughout the body. I think you probably already know the locations of most of the adipose. I don't think this is a very good list anyway, because it just lists areolar tissue. That's not very helpful. So if you think about where most of your fat reserves are, what your book just lists as areolar tissue, they're talking about the hypodermis under your skin, which can store a lot of fat. The mesenteries, the main one is the greater omentum, which is the one we call the fatty apron, which gives people their beer guts. But one thing they don't mention with the mesenteries is that some fat is stored in all people in the visceral membranes, in those mesenteries, and we call that visceral fat. And visceral fat, when it becomes excessive, tends to trigger inflammation. And this is way beyond where we're going with this. But people who have problems with obesity, the adipose tissues in their mesenteries, that visceral fat, tends to trigger inflammation. And that creates secondary problems beyond just having the fat tissue there. All right, the third metabolic tissue that we're looking at is skeletal muscle. Skeletal muscle does maintain glycogen reserves, but that glycogen reserve is going to be used by your skeletal muscle. So you really don't break down that glycogen reserve to feed the rest of your body unless you're really just like starving. So just overnight, that's not long enough to take the glycogen reserves out of your skeletal muscle. But if you are in a prolonged calorie-restricted diet, whether it's voluntary or involuntary, you'll not only release that glycogen, you'll actually start breaking down your muscle protein and releasing the protein to feed the rest of your body. Remember, your main task is to keep enough glucose to your brain. So the nerve tissue itself doesn't maintain its own reserves, and so it requires two absolute things from the bloodstream at all time: glucose and oxygen. So if you get hypoglycemia, if your blood glucose level drops too low, then your nerve cells just shut down, and you'll actually lose consciousness really rather abruptly if it drops too low. Okay, so the rest of your tissues, we're talking about connective tissue cells and epithelial cells, glandular cells, whatever. They are just going to run off what they can get in the bloodstream. They're going to have glucose if you just ate. Otherwise, they're going to feed off fatty acids. And that's going to depend on hormones, the hormones of the metabolic states. And we're going to talk about those. So in the absorptive state, that's when you have nutrient absorption underway from the gut. Your book says it lasts for about 4 hours. Well, of course, that depends on what you ate. If you just ate a few crackers or dry toast, it's not going to last that long. So we're talking about a typical meal that has a balance of carbohydrates and lipids and proteins. The post-absorptive state is when your gut is empty. I kind of divide it into two periods, the immediate and the prolonged post-absorptive state. The longest you tend to go in the post-absorptive state is if you ate dinner at, say, 6 in the evening and you didn't eat again until breakfast the next morning at 6. So assuming you ate a big dinner and your absorptive state lasts about 4 hours, well, then maybe you're post-absorptive for 8 hours. So that's about as long as most of us tend to go in the absorptive state. So, but if you skipped a meal or if you are just not eating or you're starving, whatever, then you're going to be in that prolonged post-absorptive state. And the difference there is you add stress hormones on top of just the hormones of the typical post-absorptive state. So if you're post-absorptive, remember your brain's going to get all your glucose, and everybody else is going to rely on lipids. And it says amino acids, but I don't think most cells can break down amino acids. All right, so here you just see a little map of the different glucose levels. Notice that, or not just glucose, lipids, and amino acids. And of course, it's going up right after a meal, and then it's dropping. So maybe by noon, you are starting to get post-absorptive, but then you eat again. And maybe by 5, you're starting to get post-absorptive again, but then you eat again. So most of us are not in the post-absorptive state if you eat regular meals, breakfast, lunch, and dinner. But overnight, then you see that you can be in the post-absorptive state for maybe 8 hours or so, 8 to 10 hours. All right, so here we are looking at the absorptive state. And you can tell that because at the very top it says blood glucose levels are elevated. And notice that we also have insulin here. Insulin is the hormone of the absorptive state. When glucose levels go up, the pancreas secretes insulin. Insulin activates membrane transporters in typical body cells and allows the intake of glucose. It doesn't show it in this picture, but it also allows the intake of amino acids in typical body cells. Now, I say typical here, we're talking about muscle, epithelial, and connective. Your liver is special, your brain is special, and your adipose tissue is special. So the liver can pick up glucose without needing insulin to turn it on. What insulin does in your liver cells is it actually activates the formation of glycogen, our storage form. And your brain cells do not need insulin. Brain cells get to eat glucose whether you're in the absorptive state or not. So you're going to take in not just glucose, but also amino acids. Your lipid level is going to be high. Look on the left. And blood amino acids over there on the right, it says insulin and growth hormone help pull those in. So basically, you're putting the groceries away, and you're stockpiling. You're doing lipogenesis to make triglycerides in your cells. You are storing as glycogen if it's a cell type that can store glycogen, and again, that's skeletal muscle and liver. And then you're going to make proteins depending on what the hormone signals are telling you. So if you're a kid and you're growing, you're going to secrete growth hormone, and as long as you eat, that growth hormone is going to stimulate protein synthesis. But if those proteins, whether it's a kid with growth hormone or pubertal teenagers with sex hormones, whatever, you've got to have the insulin there for those other hormones to stimulate protein synthesis. And at the bottom of the figure, you can see, yeah, I'm going to burn sugar. I'm mostly going to burn sugar because that's mostly what I have. I'm going to stockpile my triglycerides. I'm going to make my proteins. And because my blood glucose level is elevated and my insulin is letting me take it in, my cells are going to be able to burn glucose and stockpile everybody else. Okay, so now we are in the post-absorptive state. The glucose is coming from the liver. There's no insulin in this picture. The hormone of the post-absorptive state, not shown in this picture, is glucagon. Glucagon is secreted by the pancreas when you're in the post-absorptive state, when glucose level is low. And glucagon, there I see it under carbohydrates. Glucagon stimulates the breakdown of glycogen to glucose. Again, that's primarily in liver cells. And triglycerides are going to be broken down as well under the stimulation of these hormones, including glucagon. Now, you see epinephrine and glucocorticoids listed there as well. Those are stress hormones. So they're going to be activated in the prolonged post-absorptive state. So looking at the overview, I'm going to burn my triglycerides, I'm going to burn my glucose, I'm going to burn my proteins. I will only burn proteins if I'm in the prolonged post-absorptive state and I'm in dire need of more nutrients. If I have enough fat reserves, I'm not going to break down those proteins. And you'll notice that the glucocorticoids, again, are involved in protein digestion, so that's when you're stressed. Okay? Glucocorticoids are hormones of stress, as is epinephrine. All right, so that's kind of the overview. Glucagon is going to signal the liver, period. And the stress hormones can signal adipose reserves or skeletal muscle cells and make them break down in dire straits when you're stressed. At the very bottom, notice that you're going to release ketone bodies. Ketone bodies are released again in that prolonged post-absorptive state, and it occurs when you have too much of an overstimulation of these pathways. So stress puts this process into overdrive, causes the liver cells to release ketone bodies. Hey, ketone bodies are not in and of themselves a bad thing. Ketone bodies can be used for fuel by other cells. So your peripheral body cells can use ketone bodies. They can feed off ketone bodies the same way they feed off of fatty acids. But when you have, when you're in a state of profound starvation, or if you have a hormonal problem, then the ketone bodies can build up in the bloodstream. They are acids. They lower the pH. So when you have ketosis, that's too many ketones in the blood, then you can have ketoacidosis. And this is a problem in unregulated diabetes. Okay, so in the post-absorptive state, we're going to break down our lipids and amino acids. We're going to produce those ketone bodies, and it can be produced not only by fatty acid, but even by proteins. And some people go on ketogenic diets where they're trying to starve themselves to lose weight, and their whole goal is to increase the ketone body production. I'm not sure that's a good idea. You certainly can't do it for a very long period of time. So you don't need to remember the names of the 3 types of ketone bodies, but the liver cells don't eat ketones, they produce them. So basically they're feeding peripheral cells the ketone bodies, and that's going to cause ketosis if you overdo it. And so ketonemia, high levels of ketone bodies, and ketoacidosis if it goes on too long, and that can be dangerous. You can get into a state of the acidosis may be so severe that it causes suppression of neuronal activity, which can affect the brain and the heart. And in fact, in untreated diabetes, this is not an uncommon cause of death. Okay, so you're in homeostasis, I'm assuming, because you're eating regularly, you have the nutrition you need, you have a balanced diet. Malnourishment or malnutrition means you have a nutrient imbalance. So I'm not going to get bogged down in my plate and the USDA guidelines. If you're interested in reading about that, you go right ahead. By now, you probably ought to know that your diet should consist mostly of fruits and vegetables, and probably more vegetables than fruits. When we say grains, we mean whole grains, not highly processed grains that have nothing but starch left. You have to have some sort of protein. You do not need dairy products, but they're not bad for you. So there's your little my plate. And I think by now that's actually kind of, it's not necessarily out of date, but there are certainly other ways of looking at your nutrition. Okay, so back to this idea of essential. Essential nutrients, remember, ones you have to eat, you can't make. So a complete protein is one that has all the essential amino acids in sufficient quantities. And this is found in, look at all the list there, everything's animal product. Beef, fish, poultry, eggs, and milk. And they could have put pork on there. I don't know why they didn't. But incomplete proteins are what you get from plants. So if you want a balanced diet and you don't want to consume animal products, you have to get the right kind of plants, because some plants are high in one amino acids and deficient in one essential amino acid, and others have a deficiency in others. And so what we know is that if you eat a legume, which is beans and peas, and a grain, which could be corn or rice, then you're getting complete protein. So if you look at the basis of diets in indigenous populations, at least the ones I'm familiar with, it's always in Central and South America, and in Mexico, it's beans and corn, or beans and rice. And the reason you always have the bean and the grain is because that gives you complete protein. So nitrogen balance is simply a measure of how much protein am I consuming versus how much am I actually using to build protein in my diet. Because you don't store nitrogen. You don't make protein to stockpile it. You take the amino groups off and throw them away as urea. So nitrogen balance is measured by how much did you eat versus how much did you throw away. And those nitrogen-containing compounds that are waste products are going to be lost in the urine. So as we get into the urinary system, we'll look specifically at the nitrogen-containing wastes, the non-protein nitrogens that are lost in the urine and feces. So nitrogen-containing compounds that you keep and use, amino acids, purines, and pyrimidines, which you remember are used to make DNA and RNA, creatine, which is used in your muscles, and porphyrins, which are what hemoglobin is, and some of the cytochromes. Okay, so you've got to have nitrogen in your diet. These are all things you have to make, but you don't store it, so you can't stockpile nitrogen. So you're in positive nitrogen balance if you have all the nitrogen you need and you're absorbing more than you excrete. You're in negative nitrogen balance when what you excrete is greater than what you eat. And that could just be because you just eat more meat than you need. All right, minerals. You can't make these. You have to eat them. So all minerals are essential. You need some basic minerals. Some of these you're already very familiar with, sodium and chloride. Potassium, so sodium and potassium to maintain membrane potentials. Calcium you need for your bones, but you also need calcium for your muscles and for blood to clot and for exocytosis of neurons and contraction of muscles. You need iron to carry oxygen in your bloodstream. All of those are examples of the minerals that you need. So the bulk minerals, the ones you need in large amounts, because you metabolize them so much or you use them so much: sodium, potassium, chloride, and calcium. Phosphorus, which remember makes the phosphates, which are the P's of ATPs, and you need phosphate for DNA and RNA. And magnesium is usually used as a coenzyme, so but you need a lot of it. Trace minerals are things you need in smaller amounts, and some are more probably important than others, like iron. So you know you need iron for transport of oxygen. The others on that list are all components of either enzymes or coenzymes, and I'm not going to ask you to memorize what does what. How do you have reserves of these? You store calcium and phosphate in your bones. You store, that's about it. You store calcium and phosphate in your bones and iron in your liver. Okay, vitamins. These are essential organics. In other words, they're organic compounds that you can't make. You have to eat them. And vitamins for you are not necessarily the same as vitamins in other animals. For example, we need to eat vitamin C. We can't make it. Dogs and cats, no problem. It's just the primate group, monkeys and monkeys to humans, that for which vitamin C is essential. But we get a lot of it in our diet, so in general it doesn't hurt us. There are two groups, and we divide them into fat-soluble and water-soluble. The fat-soluble vitamins are A, D, E, and K. You're going to absorb them with the other lipids in your diet, which means if you don't get a lot of lipid in your diet, you may have a poor absorption of these. Other sources of vitamin D, of course, you make some of that in your skin. Usually that maxes out at 20%, so you still have to get 80% from your diet. Good sources of vitamin D include cold water fish. And vitamin K is found in things like broccoli, but also you have intestinal bacteria to produce some of the vitamin K that you need. Vitamin A is an antioxidant, and it's used to make the pigments in the retina of your eye. Vitamin D is the precursor to that hormone calcitriol that is needed to absorb calcium for all the needs of your body, so you need it for bones. Vitamin E is another antioxidant. Vitamin K we need for clotting proteins. The liver uses it to make the clotting proteins. So do you store any of these? Well, with the fat-soluble ones, you could store it, but you don't usually. The only ones I know of that people have had hypervitaminosis of is vitamin A and vitamin D can be stored in liver. So if you eat a lot of liver, you're probably getting more A and D than you need. But it's really, unless you're taking like huge supplements, it's really hard to get more than you need. And if you're eating a regular diet, you're not going to be low. Now, there are a lot of people who take vitamin D as a supplement. Some doctors advise it for older individuals. But the only way to know for sure whether you need it or whether you're absorbing it from a supplement is to do blood tests. So unless you had a blood test that said you were low on vitamin D, then probably chances are you don't need it. Our water-soluble vitamins, again, the B vitamins are coenzymes. Vitamin C is an antioxidant. And the thing about the water-soluble guys is if you eat more than you need, you just excrete it in the urine. So any vitamin excess, if you're taking vitamin C pills or you're taking multivitamins, if you're getting more of the B vitamins or vitamin C than you need, you're just peeing out whatever's left over. And so you're not going to get a hypervitaminosis because you don't store it. Bacteria in your intestine are supposed to be there. They're contributing to your health. And they produce some of the B vitamins as well as vitamin K. The only vitamin that you have trouble with is B12. And B12, which contains cobalt, is a really large molecule. And your stomach would digest it, or you wouldn't even absorb it if you didn't take extra steps. So we secrete something called intrinsic factor. This comes from the parietal cells and the gastric glands of the stomach. It binds to the B12, it protects it from being digested, and it also promotes its absorption further down in the intestine. All right, so that's just a quick run through of the vitamins and minerals and the other nutrients. We're going to shift our attention now to thermoregulation and metabolic rate. So you have what we call a basal metabolic rate, or BMR. Your basal metabolic rate is determined by your weight, your percent body fat, their genetic factors, but basically your metabolic rate is a measure of how many calories you expend. Obviously, if you exercise more, it's going to go up. So generally, we measure what we call a resting or basal metabolic rate. You gain energy when you eat. You can release that energy when you digest things, break chemical bonds. You use some of that energy to synthesize ATP, but even as phenomenal as your mitochondria are, harvesting ATP out of glucose and fats, mitochondria still lose about 60% of the energy in those chemical bonds, and that energy is lost as heat. So to measure energy, we have a standard way to measure it. It's 1 calorie is the energy required to raise the temperature of 1 gram of water by 1 degree Celsius. A kilocalorie is obviously similar to that, but it's to raise the kilogram. And a kilocalorie is the same as calorie with a capital C, and that's what we use when we're listing that information on nutrition labels. So calorimetry is simply a way of measuring that. There are different ways of doing body calorimetry. You can't do it at home. You have to have a special device. They sometimes have an entire room where they sit you in the room, and you're sitting very still, and they're just measuring the temperature of the room. And so your metabolic rate can be determined by how much the temperature of the room increases while you're sitting still in it. So that's just not a very simple thing to do. We can do calorimetry of foods. We do that by burning them, which we're not going to do to people. So lipids release about 9 calories, carbs and proteins about 4 each. So lipids have more than twice as much stored energy as either proteins or carbs. So we're going to measure your metabolic rate based on this, this loss of heat, and that can be converted to calories. And of course, it's going to be affected by a lot of different factors. And here you can see just a change in your caloric expenditure based on activity. So your basal metabolic rate is that your total resting function. And again, to be able to actually measure that, the other way to do it is to put a mask over you and breathe through a ventilator that measures oxygen consumption. Okay, so never mind measuring it, how do you regulate it? So the most important hormone in regulating metabolic rate is thyroxine. You remember this is the thyroid hormone, T3, T4. CCK is, and ACTH, these are hormones that suppress appetite. There are others. Leptin is a hormone fairly recently described that is released by adipose tissue when you're in the absorptive state. So we think it's an appetite suppressor. And ghrelin is from your stomach, and that stimulates appetite. Those appetite stimulators and suppressors are really not so much regulating your metabolism, they're regulating how much you eat. And of those, I would say, if you can remember that leptin is going to reduce your appetite and ghrelin is going to increase it, and obviously leptin during the absorptive state and ghrelin during the post-absorptive state, and then thyroxine controls your overall metabolism. There are so many other hormones they found in addition to CCK that suppress appetite. We're not even going to get started on those. All right, so obesity is, it's a medical term, and it has a specific definition, 20% above your ideal weight. But of course, ideal weight, that is not necessarily something that you can say is a hard and fast thing, because the ideal weight for someone who is 5'6", but has very thin bones, fragile bones, is not going to be the same as the ideal weight for someone of the same height but with strong bones. So it's not that simple. The obesity epidemic in this country and in other countries has been a cause of alarm for decades now. We've been describing it, but we've been trying to figure out what's causing it. And the associated diseases going with it, heart disease, cancer, diabetes, some of these are inflammatory diseases, and we're trying to figure out how to get control of the inflammation, but just trying to figure out why we are more obese than we used to be collectively as a culture is not that straightforward. Some of it is we're just not as active as we used to be. Some of it is that we're consuming more artificial sweeteners and high fructose corn syrup. There's definitely evidence that that is a factor. And some of it is just the processed foods that we eat. So it's hard to pin down one factor as a cause of obesity. All right, thermoregulation is the mechanisms you use to keep your temperature within an acceptable range. This is hormonally controlled to some extent. It's genetically controlled to some extent. The environmental conditions are going to change what you're actively doing to maintain, but it's important to keep yourself, of course, in the temperature range that you're adapted to. So we know that heat is a byproduct of metabolism, and so the more physically active you are, the more heat you're going to generate, and you've got to lose that heat to the environment. One of the things that thyroid hormone does is it actually causes mitochondria to, they describe it as wasting ATP, just spilling ATP out and releasing the energy from it. Well, that just makes you generate more ATP, but that's fine because you generate heat when you do that. So thyroxine specifically increases the temperature of the body. So heat exchange, the way we lose heat, radiation, convection, evaporation, and conduction. Radiation is radiant energy. You're actually radiating heat as infrared rays. And how well you can do that depends on how much skin you have exposed to the environment and how different the environmental temperature is from your skin temperature. Convection is the heat lost to air, so the air warms up as it hits your body surface and then leaves, taking some of that heat with it. And then evaporative heat, of course, this is from sweating. So as water molecules evaporate off the surface of your skin, they take heat with it. About 20% of that is insensible. Sensible perspiration means you've got sweat trickling or collecting on your skin. Insensible, your skin doesn't feel wet. And then conduction is if you're in physical contact with something cool, then you're actually going to conduct heat from the body. So if you're hot and you're leaning against a nice cool surface, you're doing conductive heat loss. Okay, so there are the different ways that you lose heat, and that is going to be controlled by the hypothalamus. So hypothalamus is keeping track of your temperature. The hypothalamus is keeping track of almost all of your homeostatic mechanisms. And so you have two little centers. One's going to turn on, let's gain heat. The other one's going to turn it off, let's lose heat. And you already know the main ways that you control that. You sweat, you vasodilate if you're hot, you vasoconstrict if you're cold. Behavioral modifications, put on a sweater, turn up the thermostat. So you have a set point, and that set point can vary at different times of the day. It's usually highest and therefore you're warmest at about 9 in the morning and 9 in the evening. And it dips to a low at about 3 in the afternoon and 3 in the middle of the night. So if you find yourself waking up at 2 or 3 in the morning cold and you pull an extra blanket over, you can blame your hypothalamic set point for that. Or if you get sleepy at around 3 in the afternoon, you can blame your hypothalamic set point for that. And if you're most awake and energetic at 9 in the morning or in the early evening, then you can thank your hypothalamic set point for that. Okay, hypothermia, obviously you're way too cold. That's prevented for the most part by the mechanisms that the hypothalamus initiates. Vasoconstriction, so you cut down on blood flow to the skin and reduce heat loss. Shivering, and that works most of the time, but obviously if you're in an environment that's too cold or if those mechanisms don't work well in you, then hypothermia can be a threat. Hypothermia in infants, actually in infants they have another mechanism. They have a little patch of what we call brown fat in between their shoulder blades, which is pretty well dissipated by the time you're an adult. And that generates heat before an infant is old enough to start shivering. Infants don't shiver. So it's hard to tell if they're cold just by looking at them. You've got to keep them warm, but not too warm. And in the elderly, very often the mechanisms simply don't work as well. So the individuals that are usually lost to us in a cold snap are going to be the very old, and likewise they have similar problems with getting rid of heat, so they tend to have hyperthermia as well. So we're going to have sympathetic, that's a stressor to be in a cold environment, sympathetic reactions. And of course you can get up and be more active, right? So if you're cold, you get up and walk around, jump up and down, do some exercise, then you can increase the amount of heat being produced. There's some evidence that brown fat does work in adults. I've seen some studies that say if you just turn your thermostat down a few degrees, even if you're just sitting around, if you're cooler, you tend to burn more calories. So it's a way to increase heat loss, increase calorie loss, and maintain a body weight by just turn the thermostat down. All right, so in a warm environment, you're going to vasodilate, and in a cool environment, you're going to vasoconstrict. And what we're looking at in these two pictures is those superficial veins, which you learned already are running close to the skin, and so they're able to, let's go back. When you're hot, you dilate the superficial veins. When you're cold, you constrict those and dilate the deep veins, and that way you can thermoregulate. So there's a heat transfer between warm and cool blood. So in your limbs, particularly in the lower limbs, where the artery runs up against the vein, the heat can be transferred from the artery to the vein so that the blood is cooled down as it's going to your feet, but it keeps your trunk warm. And we're going to see the same countercurrent mechanism in other settings. But this countercurrent heat exchange in the blood vessels of limbs is found in definitely in animals that are adapted to cold environments. For example, penguins, their feet very cold. They hold that heat in the core of the body through this countercurrent heat exchange mechanism. Okay, and of course we have shivering, thermogenesis. That's going to increase the contraction of muscles, which spends a lot of ATP. And non-shivering thermogenesis, this is what is going to hit those mitochondria. So mitochondria are going to be stimulated by thyroxine, and that causes them just to sort of waste ATP. You're going to increase the stress response, epinephrine, which increases the breakdown of glycogen. Anything that increases your metabolic activity increases your body temperature. Okay, you can get used to things. You can acclimatize. You get used to your environment over time. And again, there are going to be genetic differences, there are going to be body size differences, but some people are just more used to the cold weather. We live in Texas. We don't really like cold weather that much, but if you go further north, and what to us is very cold at 50 degrees, that's considered a balmy day up in Minnesota. So you do get adaptation or acclimatization over time. Your surface to volume ratio certainly makes a difference. Little people lose more heat from their much smaller core body than big people do. Especially for kids, you need to keep their heads covered because they're going to lose a lot of heat through the scalp otherwise. Their heads are just relatively big compared to the rest of their body. And here's the story on brown fat. Brown fat is a specific kind of adipose tissue that's modified to have a lot of mitochondria. That's why it's called brown. It says it's rich in heme, cytochromes, and mitochondria. It's mostly in the mitochondria. And they're gonna break down fat, and just that's gonna generate more. It's just a higher metabolic rate. It generates more heat. So very definitely in humans, but a little bit very definitely in humans and infants. But even in adults, you've got some brown fat, and there's some evidence that you can actually increase the amount of brown fat you have by just keeping your temperature cooler, keeping your environmental temperature cooler. A fever is a mechanism to offset bacterial infection. Bacteria don't do very well at higher body temperature, but of course our cells can increase their metabolic rate in a higher temperature. So our cells can tolerate a higher temperature than the bacteria can. That's kind of the underlying adaptive mechanism of fever. It's not always pathological, but could be just a response to stress, for example. And there are other things that can cause it. Pyrogens are chemicals that induce fever. Bacteria and bacterial components are pyrogens, but they're not the only things that are. So thermoregulatory responses in adults obviously varies based on your body mass, tissue distribution, the different setting of your hypothalamic thermostats, and just, how much body fat you've got. That's going to make a huge difference. Temperature oscillations in women. Estrogen regulates thermoregulatory responses, so after menopause, the lack of estrogen causes spikes in temperature. This is why postmenopausal women experience hot flashes. Okay, that's the end of this presentation. Go back through the review questions, use the review sheet to answer those questions. That's what you need to know for the test.